Bead loading is an inexpensive assay for loading membrane-impermeable particles into live adherent cells. This protocol has proven extremely useful for loading probes for single-cell or single-molecule fluorescence microscopy experiments.
Bead loading allows researchers to quickly load all sorts of molecules, including proteins, DNA, RNA, synthetic particles, or a combination of these simultaneously into single cells. When first performing bead loading, it is important to test out the tapping force. Cell lines respond differently to bead loading, and so the number and force of the taps can be modified for optimal cell loading while minimizing cell peeling.
To demonstrate the procedure, Matthew Saxton and I will be joined by our colleagues, Dr. Amanda Koch and Gabriel Galindo.
Begin by measuring approximately 5 milliliters of glass beads in a 50 milliliter conical tube. Add 25 milliliters of 2 molar sodium hydroxide to the tube. And mix gently for 2 hours using a shaker or a rotor. Spin down the tube of beads briefly in a centrifuge if the beads are in suspension. Then decant the sodium hydroxide while retaining as many beads as possible. Wash the beads thoroughly with cell culture grade water until the pH is neutral, and decant the water each time. Use a pH test strip on the effluent to confirm a neutral pH. Then wash the beads thoroughly with 100% ethanol 2 to 3 times, decanting the ethanol each time.